So nice. So I'm going to be talking about my dissertation work titled Motor Sequence Learning Elicits Mu Peak Specific Corticospinal Plasticity. So we learn new skills and fine-tune the ones that we've already learned every day, from typing on a keyboard to playing sports and games. The sensorimotor cortex is the central node within the larger network that controls motor skill acquisition, which includes regions of the brain like the premotor cortices, the prefrontal cortex, the posterior parietal cortex, the cerebellum, and even deeper structures like the basal ganglia and the hippocampus. Given the significance of motor learning and control in humans, the larger allocation of the brain regions for this function is very important. And neuronal populations in the sensorimotor cortex exhibit oscillatory activity that reflects synchronous firing of neurons in the underlying regions. For instance, here, this trace is a population level recording, where the one below here is single unit recording. So each row is a neuron, and each line there is its firing. And when you can see that whenever these neurons fire synchronously, you would see a positive deflection in the population level activity and a negative deflection when the firing is asynchronous or absent. The mu rhythm in the sensorimotor cortex is the most spectrally dominant rhythm, and it oscillates in the 8 to 13 hertz frequency range. Understanding how these neural oscillations interact with the motor learning system is critical to understand the fundamental neurophysiological mechanisms underlying human motor learning, and also to develop interventions for motor rehabilitation. The sensorimotor mu rhythm, like I mentioned, has oscillatory activity, and it reflects information processing during these brief windows of excitation and inhibition. In primates, single neuron firing rates in the motor cortex are highest during mu rhythm trough phases and lowest during mu rhythm peak phases. So evidence for this comes from a 2011 study where they recorded single neuron firing rates from M1 in non-human primates, and information of which is plotted here with the normalized firing rate along the y-axis and the phase of the alpha bin on the x-axis. And you can see here that the highest firing rates correspond to trough phases, and the lowest firing rate correspond to peak phases. In humans, corticospinal tract output and communication between M1 regions in both hemispheres are also high during mu rhythm trough phases compared to peak phases. Now, before I talk about the mu rhythm and its relationship with corticospinal tract function, I'd like to briefly talk about the corticospinal tract itself. This is the largest descending tract that controls voluntary movement. Crudely, the information traveling down the motor cortex via the corticospinal tract, and it innervates the muscles for voluntary movements. And we can measure corticospinal tract output using TMS. TMS stands for transcranial magnetic stimulation, and it is a non-invasive brain stimulation technique that works on the principle of electromagnetic induction. So you can see the TMS coil here, which has insulated copper that is wound underneath. And when you transiently pass electricity through this, it creates a magnetic field, which in turn creates an electric field that depolarizes the cortical level neurons. This stimulation depolarizes the neurons, and that travels down the corticospinal tract all the way down and innervates the alpha motor neurons at the spinal cord, which in turn innervates the peripheral nerves that control the muscle. And once you stimulate the cortical region that around the hand region, you would see what we call a motor evoked potential, an involuntary twitch in the muscle. And the amplitude of this MEP shows is a measure of corticospinal tract output. And a greater MEP amplitude would indicate a greater corticospinal tract output. And note that TMS can also be used as a tool and an intervention. And I would go into details of this as we progress. I spoke about the mu rhythm and its role in the cortex. I spoke about the mu rhythm and its role with the corticospinal tract output. And now I'm going to talk about mu rhythm and its phase influences corticospinal plasticity. So we know from TMS interventions targeted to M1 that show differential effects when delivered during mu rhythm trough phases as opposed to mu rhythm peak phases. So when these interventions were delivered during trough phases, you see long-term potentiation-like synaptic strengthening, whereas those when delivered during mu rhythm peak phases shows long-term depression-like weakening. So what do I mean by these LTP-like strengthening and LTD-like weakening? So I think a good way to think about this would be the Hebbian plasticity principle, where neurons that fire together wire together. So when the presynaptic neuron fires before the postsynaptic neuron, it causes transmitter release and strengthens that synapse. So we would see higher amplitudes after, as opposed to LTD or weakening, where the postsynaptic neuron fires before the presynaptic neuron, causing it to weaken. Evidence for this comes from a 2018 paper where they timed TMS interventions to be delivered during mu rhythm peak phases, like you can see here, or mu rhythm trough phases, like you can see in the blue line at the bottom. So they measured MEP amplitudes, presented on the y-axis across time on the x-axis. So they measured MEP amplitudes at baseline at 2 time points, after which they delivered a repetitive transcranial magnetic stimulation intervention displayed using this gray box here. And what they noticed after the stimulation, when they measured at frequent 15-minute intervals, that when the stimulation was delivered during mu rhythm trough phases, they saw an increase in MEP amplitudes, indicating LTP-like plasticity, and a decrease in MEP amplitudes when they were delivered during mu rhythm peak phases, indicating LTD-like plasticity. And this was the initial experimental evidence for corticospinal plasticity induction being associated with sensorimotor mu rhythm phase. It's also important to note here that this LTP-like mechanism is also the one that supports human motor learning. Furthermore, TMS, when applied during mu rhythm trough phases, also boosts motor learning. And evidence for this comes from a 2021 study where participants practiced an explicit sequence task using their non-dominant hand, and after which they received mu phase dependent TMS that were targeted specifically to either peaks or trough phases, and a control group also received sham stimulation. And on day 2, they were tested on the task for fewer trials. So on day 1, participants from all 3 groups learned, or rather practiced the task very similarly. Here you can see the number of correct sequences along the y-axis and the trial along the x-axis. You see that when they start, they have fewer correct trials, and eventually by the end of the learning session, they are all around 7 to 8 trials, correct sequences. After which they receive mu phase dependent stimulation, here around 1,800 pulses at 1 Hertz rate. Interestingly, it's only the group that received trough phase specific TMS intervention showed the greatest improvements in offline learning. So it's also important to note here that although the sham simulation and the peak simulation showed similar levels of improvement. So this study indicated a potential endogenous motor memory formation mechanism that is potentially biased towards mu rhythm trough phases, and that these plasticity mechanisms that were induced by TMS could reflect mu phase dependent memory formation mechanism. And although this study differentially modulated the motor memory consolidation process, it was interventional in nature, but not physiological. But we were interested to see if motor learning relies on a mu phase dependent mechanism. And to do that, we asked this question: does motor learning rely on sensorimotor mu phase dependent mechanism? So given that the mu rhythm trough phases are associated with interhemispheric communication, higher corticospinal output, improved LTP like corticospinal plasticity, and like I mentioned, it boosts motor learning. If the answer is true, then learning-induced corticospinal plasticity should vary significantly across mu phases. And we hypothesized that mu sequence learning will elicit corticospinal plasticity that would be more evident when measured during trough than peak phases. To do this, we designed an experiment using a well-established serial reaction time task. So the way this task works is that briefly, the participant is seated in front of a computer that displays 4 hyphens, right? And at one point, one of the hyphens turns into a circle, which is a cue for the participant to press the corresponding key on the response pad using the corresponding finger. So in this case, if the second hyphen turns into a circle, the participant is expected to press the second key using their middle finger. And here is a depiction of the trial structure. They do this for 120 trials. So the key here is that there is a 12-item sequence that is embedded in this, the one that is shown on the screen. So participants learn this sequence implicitly over trials. One way to think about this is thinking about it in terms of playing sequential keys on your piano to produce music, as opposed to pressing random keys, right? So the reason I bring this is because the control group in this case has only random key presses. So they do not have this embedded sequence within. And the experimental design for this is that we recruited 36 subjects, divided them randomly into the sequence and the no sequence group. And at baseline, we calibrated our phase targeting algorithm, and we measured MEP amplitudes using phase-dependent TMS, like I mentioned before, during 3 different phases: during mu rhythm peak phases, mu rhythm trough phases, and mu rhythm random phases. After this was done, both participants in both groups performed the serial reaction time task. The key difference here again is that the sequence group performed the SRTT task with this embedded sequence, whereas the no sequence group performed this similar task without any sequence in there. So they had random key presses. And after sequence learning occurred, we measured MEPs immediately and 30 minutes after the sequence task. And 1 hour from when they had completed the serial reaction time acquisition, participants also performed this task again for fewer trials, in this case, 5 blocks, to evaluate retention. And finally, they finished with the process dissociation procedure. This is a task that is used to test explicit knowledge of the sequence. So before we go into looking at the behavioral and the physiological results, I want to walk you through how this phase targeting occurs and how we evaluate if our phase targeting occurred accurately. So here you would see raw and filtered brain activity patterns from the sensorimotor activity. So this is filtered in the mu rhythm, that's 8 to 13 hertz. And here the black line represents the time when a TMS pulse was triggered. And ideally, we want this line to coincide with peaks for the orange trace and troughs for the blue trays and random phases for the green trays. And that's precisely what we see for both the sequence and the no sequence group. And similar information is just represented in terms of a polar histogram. You're probably familiar with regular histograms. The only difference between a polar histogram is that it's plotted on a polar coordinate going from 0 to 360 degrees. And each bar here represents the number of pulses. And we would expect most number of pulses to be delivered around the 90-degree direction for the peaks and around 270-degree direction for the troughs, and should to be dispersed randomly for the random groups. And that's precisely what we see here, that the clustering is around 90 for peaks, 270 for troughs, and distributed randomly. Furthermore, here I show the brain activity that was recorded from, again, the sensorimotor region. And here we see that the higher brain activity recorded during peaks, low brain activity recorded during troughs, and not so much during phases. So this shows us that our real-time targeting of the sensorimotor mu rhythm phases was accurate in both groups. Now we can go into the behavioral results. So given the task and the two groups, subjects in the sequence group who had an embedded sequence in the task showed sequence-specific improvements in reaction time during acquisition, and they also retained it 1 hour after learning. So I'd like to draw your attention to the figure here with the mean reaction time that's plotted along the y-axis, along with their block number on the x-axis. And dotted lines here represent random blocks, so in which there is no embedded sequence. For the sequence group, blocks 3 to 7 are the ones that contain the embedded sequence, and likewise for blocks 9 to 13. However, for the no sequence group, essentially every block here is a random block. So if you were to follow me with the green line here, you would see that the participants in the sequence group perform similarly during the random blocks, and their performance improves when they experience an embedded sequence. And when they are met with a random block, their reaction time worsens, and the reintroduction of a sequence into their blocks improves their performance further. So it brings their reaction time down as they're able to predict, because they're learning this across trials. Finally, we throw them off again with a random block to see if the improvements in reaction time are specific to the sequence. And the participants in the sequence group also maintain this during the retention testing, that is 1 hour after, and the reaction times go way further down. And again, with the random block, it comes up. The no sequence group, on the other hand, do not show improvements as much as the sequence group, where the reaction time is fairly constant throughout. They start around 350 and they finish around 325 milliseconds. And it's the same for the retention block. And the key metrics from this task that we use to measure are the acquisition skill, the retention skill, and offline changes. So acquisition skill is the difference in mean reaction time between the last sequence and the last random block. So it's this. For the retention skill, it's the similar difference in mean reaction time between block 1 and block 2, in this case, the random and the sequence. And a difference of these differences is called offline changes. And information of these three metrics are plotted here in panel B. So difference or change in mean reaction time along the y-axis for each group on the x-axis. And we see that the sequence group acquired the skill and they were able to retain it. However, there was no difference in offline changes between the two groups. Now moving on to the physiological results. First, we looked at the mu phase independent MEP amplitudes. And by that, I mean those that were recorded during random phases of the mu rhythm. And we see that these MEP amplitudes increased after SRTT acquisition in both groups. So in this figure, we see MEP amplitude plotted along the y-axis and the time of recording on the x-axis. We see that at baseline, immediately after serial reaction time task, and 30 minutes after. And we see that MEPs increased generally. We see a mean effect of time, and there was no effect group or a group by time interaction. So MEP amplitudes increase from baseline to immediately after, from immediately after to 30 minutes after, and also from baseline to 30 minutes after. So now to the most interesting part is the mu phase dependent MEP amplitude changes. And here we notice that these MEP amplitudes change differently across the phases in the no sequence group relative to the sequence group. And what do I mean by that? So here I have a similar plot with the MEP amplitude along the y-axis and the time of recording at the x-axis. For the sequence group, we do not see a phase by time interaction when we look at it, while we see that in the no sequence group that there is an interaction between phase and time. So this model that I'm plotting here is a follow-up to the original group by time by phase three-way interaction that was being driven by changes in baseline to 30 minutes after. So this indicates that the MEP amplitudes change differently across the phases between the groups. And this representation is by splitting the groups and visualizing that. But this is where we were surprised, right? Like, why are we seeing an effect in the sequence, in the no sequence group and not in the sequence group? However, the statement, what I just made, is not accurate because of this group by time by phase interaction. But if you think about it, the task that the participants performed in both groups differed only in the presence or absence of the stimulus, right? So the sequence group had an embedded sequence, while the no sequence group did not. So essentially, if we were to subtract the no sequence group's effects from that of the sequence group, that should give us sequence-specific changes in MEP amplitudes. And that's precisely what we did here to visualize this information. And here we have model fitted values along the x-axis. These are differences between the sequence and the no sequence groups. That is, again, across the time points of measurements from the plot I presented earlier. Here we see that sequence-specific learning recruits peak rather than trough-specific neurophysiological mechanisms. And here, a greater sequence-specific corticospinal plasticity is represented by much more positive value here, which is, again, this little difference that we're talking about. Finally, we evaluated the relationship between the changes in peak-specific corticospinal plasticity that we've just observed with the behavioral results, that is, the performance in the serial reaction time task that I spoke about. Here, again, although we expected a positive relationship, we see a negative relationship between peak-specific MEP amplitudes and behavioral measure skill. So here I have the acquisition and retention skill measure that I mentioned was the difference between the last and first sequence and random blocks. So a much higher positive value would indicate that the participant gained skill, and in this case, retained that skill. A lower value indicates worse skill learning. And that's precisely what we see for the no sequence group plotted in purple. And the metric along the x-axis is the change in peak MEP amplitude differences, that is between baseline to 30 minutes after learning. And we see this a really strong negative relationship, whereas no such relationship for the no sequence group. And the panel B here shows a similar plot for trough-specific MEP amplitude differences. Although we see these results that they appear to be similar, it was only significant in the sequence group for peak specific MEP amplitudes. So this group by MEP difference interaction tells us that peak specific corticospinal plasticity is strongest when motor sequence learning is the poorest. So in summary, behaviorally, the sequence group acquired greater sequence learning than participants in the no sequence group, and they were also able to retain this knowledge over time, in this case, an hour. Physiologically, both groups showed mu phase independent increases in corticospinal output following SRTT acquisition. So as a reminder, this is the MEP amplitudes recorded during mu random phases. And relative to the no sequence group, the sequence group showed greater increases in peak specific corticospinal tract output. This is the three-way interaction and the group specific follow-up models that I presented earlier. And these peak specific increases in corticospinal output were negatively associated with the magnitude of sequence specific learning, the last plot I showed. Together, findings provide first evidence that motor sequence learning elicits behaviorally relevant mu phase dependent corticospinal plasticity. So what do these results mean, right? So like I mentioned, previous studies have shown that the M1 spiking rates are slowest when measured during mu rhythm peak phases. And mu peak phases reflect brief quiescent windows during which corticospinal and sensorimotor network communication is diminished relative to mu rhythm trough phases. And they reflect a neutrally excitable state where the sensorimotor network propagates information weakly compared to mu rhythm trough phases. And evidence for this, again, comes from the 2021 study that I presented earlier, where they measured the propagation of TMS-induced changes in broadband power to be weak during mu rhythm peak phases than trough phases. Given that motor memories are consolidated during short windows of wakefulness and are also compressed, motor sequences are replayed during these windows, we think that the sensorimotor activity during mu peak phases may prevent motor sequence memories from being disrupted by task-irrelevant sensory information, such that these sequence memories can be replayed and consolidated via a compensatory protective mechanism. And a way to think about this is, say you were studying for an exam and you want to minimize any sort of interference from your, say, social media post, for instance. So you would turn off the notifications on your phone. And that's precisely what we think the mu rhythm peak phases are doing in this situation. And yep, that's the key findings from the study. And I'd like to take this time to thank my lab members, my supervisor, Dr. Sarah Hussain, and UT Austin for their facilities. And I'll be very happy to answer any questions that you might have. And if you don't want to ask your questions here, you can feel free to reach out to me with questions, or if you want to talk more about. And yeah.